Hey guys, welcome to my YouTube channel Medicoverse. I'm Shruti Muthukumar. In this video, I'm covering a very important topic for NEET, which is Mendelian diseases or Mendelian disorders. So, genetic disorders is a very important topic, and you often get many questions from the chapter Principal Inheritance. And in this video, I'm going to be covering Mendelian diseases, and I'm going to make this video in three parts. So, part one is basically Mendelian disorders covering color blindness, haemophilia and sickle cell anemia, and also I'll be talking about what is autosomal dominant, autosomal recessive, and X-linked dominant and X-linked recessive, so all these. And then the next video will be about the other three Mendelian disorders, and the last video will be about chromosomal disorders, which is a very easy part. Often all of us find it very easy. I hope you all understand. So for doing this, I made a PPT, and I also included my handwritten notes, which I made during my NEET preparation. So let's see that, and we can learn from that. That's just like a short notes from which we can learn. We just revise everything quickly. So this topic is very important, as I said. So if you want, you can take screenshots of the handwritten notes. It's very easy. That covers all the topics. So now let's move forward. So genetic disorders, as you all know, is classified into two, which one is Mendelian disorders and the other one is chromosomal disorders. So Mendelian disorders basically occurs due to mutation in a single gene, whereas chromosomal disorders occur due to absence or excess or abnormal presence of the chromosome, abnormalities in the chromosome, entire chromosome, and not a single gene. So that is chromosomal disorder, and Mendelian disorder means we are talking about a mutation in a single gene. So let's move forward. So we're going to see about autosomal disorders first, and in this is a diagram from our NCERT textbook. It is very important, and if you guys want me to make a video separately on pedigree analysis, then let me know in the comment section, because that is itself a very important topic, and I'll try to make it a very short video, and we can cover all the important points from that. Now, autosomal disorders are basically, as the name suggests, they are the disorders that are inherited through the autosomes. And here we can see two pedigree charts, and this is figure 5.4 of Principle of Inheritance chapter in our NCERT textbook. And the first one is autosomal dominant trait. Now, autosomal dominant means if suppose we take a gene, say two alleles, say capital R and small r. So if there's one capital R and one small r, the heterozygous condition, you are still diseased. Only the recessive condition, that is small r and small r, says that you are not diseased. You don't have the disease. So here we can see autosomal dominant trait. Here we can see the mother is deceased and the father is a normal, he has normal alleles. And you can see the son is deceased, and you can also see that some of the children are not deceased, which shows that, which basically shows that the mother is a heterozygous condition. So if suppose we keep it capital R and small r, she is capital R, small r, and the father is small r and small r. So easily we can predict the alleles from looking at a pedigree analysis. And one important example given here is myotonic dystrophy, which is given for autosomal dominant trait. So this is not mentioned elsewhere in the textbook. This is given in this particular diagram, under the diagram. So all these minute things are really important, because they may ask you a question from here. So which of the following is an example of autosomal dominant trait, for example, and you have to choose myotonic dystrophy if that is one of the options. So you can see one more thing. It's a dominant disorder, right? So you can this is the first, the A pedigree chart. This one is a autosomal dominant disorder. So you can see it in all the generations. So you can see this is generation 1, this is your generation 2, and this is also generation 3. But here, since both of them are recessive, you don't find it over here. But you can see this is generation 3, and you find one over here. So autosomal dominant, you can see this in all the generations in the pedigree chart. Now here, coming to autosomal recessive, for example, sickle cell anemia, which is given over here, and we are talking about sickle cell anemia in this video. So here you can see the disease is not seen in all the generations. You can see G1, generation 1, they don't have this disease, and which means both the parents, of course, are heterozygous, right? Because since two of the children, they are having the disease. So and this is recessive. So recessive means both suppose again, if we take capital R small r and capital R small r, then you should have small r and small r as a combination for the diseased phenotype. So the genotype should be small r for showing the diseased phenotype. So you can see the disease in generation 2 and generation 3. And now, as I said, the example for this is sickle cell anemia. And next, when we see here X-linked disorders. And here X-linked disorders means they are carried by the X chromosome. So here, when we say usually, X-linked disorders is quite rare, if you say, in females, because they have 2 X chromosomes. While male have only 1 X chromosome. So X-linked disorders, you see them more, especially X-linked recessive disorder, you see them more in the males. For example, color blindness. So here when we see X-linked recessive, you can see this is their affected mother. All the sons are affected from the mother, you can see. So 100% of the sons are affected. And here you can see again, sons. So basically the method to analyze if it's a X-linked disorder from a pedigree chart is to check if the disorder is spread to, like, from one generation to, for example, if it's mother in the first generation, then spread to all the sons in the second generation. So that's how you can find out easily if it's a pedigree of X-linked disorder. And you can see X-linked dominant, for which we don't have any example in our NCERT textbook, but I just included here. So X-linked dominant, you can see this affected mother, and you can see 50% of the children are affected. And from here, this example from here, you can see from the father, from affected father, all the daughters are affected. Right, so you can see from one, if it's father in one generation, then it goes to the daughters in the next generation. So that is the characteristic of X-linked disorders. Now coming to Mendelian disorders and the 3 examples that we are seeing very detailed in this video. So this is my handmade notes, and we'll learn from this. So you can take screenshot if you'd like to. And Mendelian disorders, basically, as I mentioned earlier, it occurs due to alteration or mutation in a single gene. So example of such disorders, you have haemophilia, color blindness, cystic fibrosis, sickle cell anemia, thalassemia, and phenylketonuria. Now, one by one, if we see, then first we have color blindness. So color blindness, what is really important here for all the diseases is to note down whether you have to know if they're autosomal or X-linked, and you're supposed to know if they're dominant or recessive. That is very important. And first, if we see color blindness, then here you can see it is a X-linked recessive disorder. It is an X-linked recessive disorder. That means the disease genotype, for a disease phenotype, the genotype would be recessive, right? So since this is X-linked, so it will be if it is in males, and if I take the allele for the one showing the disease, if I take it as XC, as shown over here, XC, if I take it as XC, then XCY is a disease phenotype. And XC will also be a diseased phenotype, but then that is very rare, and only 0.4% of females are having color blindness, where about 8% males are having color blindness. So you can see it is an X-linked recessive disorder due to defect in either red or green cone of the eye, resulting in failure to discriminate between red and green color. Now, if I show you this image, then you can see this is for normal vision. In normal vision, you can see that this is a red flower, whereas a colorblind person can see it like this. This is a green color, and a colorblind person perceives it as this color, and yellow, which is seen as yellow. So this is how it's seen. This is the difference. So now we saw that it's a failure of the eye to discriminate between the red and the green color, and it is a X-linked recessive disorder. Now, this occurs in about 8% of males and 0.4% of females. This is because females, as I told you, have 2 X chromosomes. So even if one is having the colorblind allele, suppose XC, and if the other one X is normal, that means they're just a carrier, but they are not colorblind. But they will pass it on to their sons. Included one example over here, as you can see. This is the mother. If the mother is a carrier, XC and X, and father is normal, XY, and XC here is the diseased allele. So XC I represent here as the colorblind allele. Now you can see XCX. So this X and this is XCX. This female, this daughter is a carrier. This is a normal daughter, and this is a colorblind son, and this is a normal son. So you can see 50% of the sons here are colorblind. And you can see daughters, they don't become colorblind, but 50% of them are carriers. And coming XCX, mother XCX, she's a carrier, and father is colorblind here in example 2. There we saw a normal father, right? So here in example 2, we are seeing that the father is colorblind and mother is a carrier. And you can see here first XC, XC, one XC from the mother and one XC from the father, and you see a affected female, which is very rare. And then here you see a carrier female, and again here you see 50% of the sons are affected and 50% are normal. And in example 3, you see a colorblind mother and a colorblind father, and you see all the offsprings are colorblind in this case. Now, these were the next coming to hemophilia. Hemophilia is basically a X-linked recessive disease, same like color blindness. Just remember, this is very important. X-linked recessive disease means, again, if I represent the allele as XH, then only XH is deceased, or XHY is also deceased allele. And if there is if it is a heterozygous condition, that is XH and X, then they are not deceased, they are a carrier. Now you can see in this disease, a single protein in a cascade of protein involved in the clotting of blood is affected. Now, only one single protein, which is part of a cascade of protein and clotting, which is a clotting factor, is affected. And as a result of that, a single cut in the hemophilic person, in the affected person, leads to non-stop bleeding. So the possibility of female becoming hemophilic, again, as we saw in color blindness, the possibility of female becoming a hemophilic is a rare condition. It's even more rare than color blindness, because here the mother should be a carrier and father should be hemophilic, who is usually unviable in the later stages of life. And one thing that we are supposed to remember here, which is an example given in NCRT, is that Queen Victoria was a carrier of the hemophilic gene, and it was passed down to all her successors. The males, many of the males, they died at those times because of hemophilia, because a simple cut would lead to non-stop bleeding, and it was only passed down to the males, and the females, they were just carriers of it. Now coming to the example, as you can see, XHX, this is a carrier, and XY is a normal father. So you can see XHX, a carrier daughter, normal daughter, hemophilic son, and a normal son. 50% of the sons are affected, and they are having hemophilia, whereas 50% of the females, that is daughters, they are carriers, and they pass it on to their sons. So this was about hemophilia. Now, as you can see in this image, what is hemophilia actually? So if we get a simple cut or anything, then while the blood flows out, you need the activated platelets and the fibrin. We have learned about this in grade 11, right? In the circulation chapter, we see how the clotting takes place. So you need a clot to be formed like this, and you need a blood clot to be formed so that the bleeding can be stopped. But in hemophilic person, since the one protein in a cascade of proteins is affected, this clot is not formed and they bleed continuously. So that is about hemophilia. And then now coming to sickle cell anemia. This is very important. You get a lot of questions from this topic. It is an autosomal recessive trait, and it can be transmitted from parents to offsprings when both parents are carrier for the gene. That is heterozygous condition. So if I represent this allele as HBS for sickle cell anemia and HBA for normal, which is the normal allele, then HBS is the deceased phenotype. The genotype for the deceased phenotype, and HBS, the heterozygous condition, is the carrier. And when both parents are having that heterozygous condition, it is passed on to the progeny, and they have HBS, which is which shows the phenotype of sickle cell anemia. And basically, sickle cell anemia is, your RBCs have a biconcave shape in general, but here they become sickle shaped, and hence the oxygen carrying done by the RBCs are not efficient. So we can see this defect is caused by the substitution of glutamic acid, GLU, by valine at the 6th position of the beta globin chain of the hemoglobin molecule. Now, this may sound confusing, but let me explain it to you with the help of the image. As you can see, this is an image of the hemoglobin, and this is beta 1, beta 2. Basically, beta and alpha over here, if you've studied the protein structure, you know that this is a quaternary structure of protein. And here, when we say alpha and beta, we are talking about the protein structure. and you learn that in biomolecules and even in chemistry and again and again. So here you can see beta 1 and beta 2. This is alpha 2 and alpha 1, and in between we have the heme group. Now they are saying that in the 6th position of the beta globin chain, that is in the beta globin, the 6th position, glutamic acid amino acid is substituted by valine, and that is a mutation that is taking place. Now if we see, this is a diagram given in the NCERT textbook, and this is a very important diagram. This is figure 5.15, and here in this image you can see this is the normal hemoglobin and this is a sickle shaped hemoglobin, and the normal HbA gene has GAG and CTC on the complementary strand. This is the DNA part, and this is the mRNA, and you can see mRNA is GAG, and the amino acid that is formed is glutamic acid, right? Now coming to the modified, the one that is mutated, that is HBsG. Instead of GAG, you can see the defect over here as GT. This is GTG. You can see here. Yeah, this is GTG, and you can see that GTG, and you can see in the complementary strand it's CAC, and the mRNA becomes GUG, and here the mRNA was GAG. Now when we look at this table which is given in the molecular basis chapter, that is which codon codes for which amino acid, we can see that right here GAG codes for glutamic acid. Let me annotate. We can say, yeah, we can see here that GAG codes for glutamic acid. GAG codes for glutamic acid, and now this mutates to GUG, which codes for valine. So here comes the difference in the 6th position of the beta globin chain. GAG changes to GUG, and glutamic acid becomes your valine, and hence you can see that a different protein is formed. Just a second. Yeah. So you can see from the substitution here, what happens is here, as you can see, it's caused by the substitution of glutamic acid GLU by valine at the 6th position of the beta globin chain. I just showed you what a beta globin chain is, and 6th position of the beta globin chain in the hemoglobin molecule. The substitution takes place from glutamic acid to valine, and this is because of the single base substitution, just a single base, that is this A. GAG becomes GUG, A becomes U. That is the only change that takes place. Single base substitution takes place, and you can also call this a point mutation, just a mutation in the single base. And the mutant hemoglobin undergoes polymerization under low oxygen tension. So, which is what happens, right? It undergoes polymerization under low oxygen tension, and the RBC shape changes from biconcave shape to sickle cell shape. So as I showed you, this is the sickle shaped, and hence it leads to anemia and all that, because it's not able to carry oxygen efficiently. And yeah, so that's it. We have completed 3 most important topics from our NCERT, and from which we get questions often, that is color blindness, haemophilia, and sickle cell anemia. Hope you all understood. If you want anything, if you want the handwritten notes, you can take a screenshot from this page, and I'll provide the PDF if you want in the description. And thank you so much for watching this video. If you have any doubts, you can put in the comment section below. And part 2 of the video with the next 3 genetic disorders, that is phenylketonuria, cystic fibrosis, and thalassemia, will be uploaded very soon after this video. Thank you so much for watching. Bye.